Poliovirus is a virus that carries positive sense single-stranded RNA and causes the disease poliomyelitis, which is characterized by paralysis of the legs and lungs. Poliomyelitis, or polio, is an enterovirus, a class of viruses named for their route of transmission through ingestion. Poliovirus can be transmitted through contaminated food or water, which infects the digestional tract. From there, poliovirus enters the central nervous system. The characteristic paralysis caused by poliovirus occurs when the virus replicates in motor neurons within the spinal cord, brainstem, or motor cortex. The replication of poliovirus results in the destruction of infected motor neurons. The mechanisms by which poliovirus enters the central nervous system are poorly understood. Three non-mutually exclusive hypotheses centered around the concept of primary viremia have been suggested to explain its entry. Primary viremia is the initial spread of virus in the blood from the first site of infection. Poliovirus infects cells containing surface protein CD155, which is found in Old World monkeys, higher primates, and humans. CD155's normal cellular function is the establishment of adherence junctions between epithelial cells, intestinal humoral immune responses, and the positive selection of MHC-independent T cells in the thymus.
Innate immunity is the part of the immune system that someone is born with. It is composed of barriers such as the skin, mucus, and stomach acid. It is considered the first line of defense and is also composed of cells such as phagocytes and certain leukocytes, which include neutrophils, eosinophils, and basophils. Other cells that are included are the macrophage, dendritic cell, and mast cell. Neutrophils are the most abundant type and use phagocytosis to destroy pathogens after responding to chemical signal from infected tissue. The other main type of phagocytic cell is the macrophage. Eosinophils defend against parasitic worms and other multicellular invaders, while histamine, which is released by mast cells, causes an inflammatory response. As part of the inflammatory response, blood flow increases, vasodilation occurs, blood vessels increase in permeability, which attracts more phagocytes. Cytokines are also released by activated macrophages and neutrophils, which attract other immune cells, as well as blood flow to the area. Some cytokines are also able to increase the body's temperature, which can cause the pathogen to be less effective. Symptoms of the inflammatory response include fevers and runny noses.
At the cellular level, cells in the digestive tract or central nervous system which have been infected by poliovirus present antigen fragments on class I MHC proteins, which are also called non-professional MHC proteins. Cytotoxic T cells bind to the class I MHC proteins via the surface protein CD8, a process which activates the cytotoxic T cell and makes it an active killer cell, capable of releasing perforin, which punctures the infected cell membrane, and other proteolytic enzymes to degrade the infected cell and prevent further infection. The binding of cytotoxic T cells to class I MHC molecules also stimulates clonal selection and reproduction, creating more effector cytotoxic T cells and memory T cells. At the humoral level, macrophages in the bloodstream take up poliovirus for digestion in lysosomes. Antigens derived from this digestive process are bound to class II MHC proteins and presented on the cell membrane of the macrophage. Helper T cells recognize the antigen and bind using surface protein CD4, which activates them and stimulates the release of cytokines, as well as initiating clonal selection and reproduction. Cytokines attract B cells, some of which release MAB-A12 and F12 antibodies, both capable of neutralizing poliovirus. MAB-A12, commonly designated A12, is an antibody which displays a pentaradial symmetry designed to bind up to 5 cellular receptors on types 1 and 2 poliovirus molecules and cause agglutination and easier endocytosis by macrophages. F12 is a cross-neutralizing antibody which aids in the agglutination process by binding to an additional receptor on the poliovirus cell. B cells that produce A12 and F12 antibodies bind to their substrates on the poliovirus and replicate, creating both plasma and memory cells. The plasma cells, also called effector cells, create more of the A12 and F12 antibodies, while memory cells are designed as a kind of template for future infections.
The secondary response occurs after someone is already exposed to a pathogen. Patients can respond quickly and antibodies can kill off the pathogen in a short amount of time. Because memory cells are pre-existing, the antigens are recognized quickly and effectively. The lag phase for a secondary response, 1 to 4 days, is a lot shorter than a primary response, 4 to 7 days. Antibodies in the secondary response show a higher affinity for antigens due to memory cells. The memory cells are created after the primary response and stay for a long time. They are able to quickly respond if the pathogen acts again.
In the late 1980s, an effort organized by the World Health Organization largely eradicated poliovirus across the world by using the formalin inactivated Salk polio vaccine, also called IPV, and the Sabin oral polio vaccine, also called OPV. OPV was the vaccine of choice for the poliomyelitis eradication program because it induces both a systemic and mucosal immune response while still being cheap to produce and easy to distribute. Romania, often considered to be the front lines in the battle against polio, was the original testing ground for the development of vaccines, and the research performed there led to the development of the two most effective and prevalent vaccine forms used today. The incidence of paralytic poliomyelitis in Romania decreased from 10 cases per 100,000 in 1949 to 0.1 cases per 100,000 by the mid-1980s by using inactivated poliovirus vaccine in 1957 and oral poliovirus vaccine in 1961. Both types of vaccine increase immune response to poliovirus by exposing the immune system to fragments of deactivated poliovirus, which stimulates the development of memory cells primed for future infection. The major risk of OPV, as with any vaccine, is the appearance of vaccine immune strains and derivatives of poliovirus, including vaccine-associated paralytic poliomyelitis cases, VAPP, and the emergence of vaccine-derived poliovirus strains.